think the one is having a problem with audio.
Yeah, it looks Supratik is having a problem because it continuously says connecting to audio.
Yeah.
We are live, sir, on YouTube. We can start, sir.
Sure. Yeah.
Right, sir.
Welcome to this live session for the course Functional Genomics. I am Ganesh, and I'm with me. We have Dr. Rashmi Parihar, who has been handling the portal and assisting you with all the answers for the queries. As you know, this is a live session, so students could join the live session and ask questions. And of course, we also have kept the option of adding the question on the Google form. Therefore, I'll be able to take up some of them and discuss. I see Smriti Sonkar. Maybe Smriti, you can introduce yourself. That would help.
Yeah, Smriti, can you hear us? If you can hear us, then you please introduce yourself.
If not Smriti, maybe Supratik. I don't know whether you're able to hear us.
Yeah, Supratik, can you hear us? So even if you can message it over this one, YouTube, then
Yeah,
also we'll be able to know that whether you are having some connectivity issue and you are able to hear us. So accordingly, we can help you.
Anyway, that's fine.
Yeah,
I think you can use the chat box in the YouTube link. Those who are watching this session using the YouTube can use that option. But let me once again welcome all of you for this live session, and then we can let me first take the questions that have already been posted. One of the question is that how I survive in industry. That is nothing to do with functional genomics. It is not about the survival. Okay. So all of us, we study different subjects, and then we go to a job and things like that, and then we read newspaper, we watch TV, and this is not for survival. This is beyond survival. Okay, let's not look at how one would survive. This is to use your potential to the maximum capacity, and then do something different. That's what we look at, and that's where we study, learn things, discover. If you're a scientist, if you're a technology guy, you're going to invent something, or you get into a profession like teaching, you want to make a difference in the way the student understand the concepts. In any field that you take, you want to do something different, therefore you can excel. That is what your aim should be. It is not to survive. So the simplest thing is one has to ask in any field that you take up, what your employer is looking at, what for they have hired you. And if in industry that you're joined as an intern, and they would like to see how good you learn things, therefore they see a potential that you can be a good learner, and you can therefore do well if you are given a job. If you are given a job, then they're going to see whether you are able to fulfill the expectations and go beyond. See, that is how it is. You have to see what they are expecting, and then how you can do well. So for that, you have to learn, you have to read, you have to think, you have to contribute, regardless which profession you take. So don't look at mere survival. Everyone survives, including all the animals and plants. But the difference in the human is that you are able to do better than the animals and plants. That's why we are ruling the planet. So think at from that angle. Okay. There's another question, which is that I'm a student of BSc. Which course in the NPTEL platform would be better after completing the course? More competent in the field of genomics. You have to look at what is that you want. I mean, there are a couple of courses in genomics, and if you're good with some bit of mathematics, then you can take up some courses in bioinformatics, computational biology, or you can go and look at cell and molecular biology courses. Things like that. These are all introductory topics. It's not like something that covers everything, like a program like MSc or BSc or BPharma or BTech. So, but these are some courses that gives you an opportunity for you to learn from experts who are there in the country, who are otherwise are not faculty in the institute or university where you are studying. So, these courses on their own doesn't make any a program, but it highlights, given you get to learn something. But the NPTEL also, if you look at very carefully, they have made some baskets. So if you take 3 courses which they recommend, you earn certain types of certificates that you want to go and explore. That's what I would suggest. That is from Ishan. So Pratik has some 2 or 3 questions. One is on epigenetics and cancer. So there are 2 ways of looking at. I don't know what you really intend to ask, whether your habits would make a difference in the genome, and that would make a difference in the way the tumor progress. If that is the question, then it's still a long way to go. But what we do know is that the cancer cell is something that is like a rebellion in your body. Okay, they don't. These cells are not under your command, and then they do things that you don't like to do. So they are out of control. They are rebellion. They are on their own, and they are able to do that because they are able to change the way the genome was being regulated in other tissue. For example, if it's a nerve tissue, then the neurons don't divide. So pretty much the genes that are involved in cell cycle are suppressed. But if it is like involved in skin, often you have damage in the skin, so it has to proliferate. So there are genes that gets activated there. So it happens because of the epigenetics change, the way the chromatin is organized, which regulates set of genes to make that particular tissue type what it is, whether it is skin, liver, whatever it is, muscle, and so on. What happens in the cancer is that they go out of this control, and it is known that the epigenetic profile also is altered. It's still a big question how that gets altered, because it is secondary, primary, one can debate. But certainly what we know is that the epigenetic profile of all these cells have been altered such that they are free of the control, and then they are able to divide and move and colonize in different sites and so on. So and people are now trying to look at drugs that would bring back the chromatin changes, and then you're able to kill those cells and so on. So this is a short answer for that. What are the latest advancements made in the field of cancer research in providing more early diagnosis? I don't know. Cancer is something still, when you diagnose it is late. This is the biggest challenge. The challenge is that how early you can diagnose it. Okay, so but you get to know that you have a cancer only when you have some form of symptoms. Okay, that you go to the doctor, and then if he or she is a really experienced doctor, then they suspect, then ask you to go for some scan and so on, like that. So it is often the first line of diagnosis is looking at your, of course, other than the parameters that they do with blood tests, they ask you to go for scanning and so on. So by then it is late, right? One can predict it much earlier is the question. But for that, it's going to be extremely difficult to model it in the human. So that's something that's why people do it in the mouse and so on. So you induce tumor and follow it through, right? What are the changes that happen? So people are trying to look at several aspects. Some of them is something it should be easy to detect regardless where is the cancer. Are there any biomarkers that you can get in the blood itself, like serum? People talk about these vesicles that come out of the cells that may have some, the RNAs, microRNAs, and so on. So this is what people are trying to really do and come up with. I don't think that we have reached a stage where you have a very good biomarker which can predict the cancer onset even before any symptoms that would come in. So that's still a long way, but this is how people are trying to look at it. Okay. Then there's another question like this CRISPR-Cas9 and siRNA. Of course, these are two different approaches. Okay, whereas your CRISPR, okay, whereas your siRNA is to silence the gene. So basically, you do not really change the gene, but you silence the RNA in the sense that it gets degraded. Therefore, there are not enough protein. It's a partial silencing. Normally, it is what is called as loss of function. You try to remove these RNAs from the cell or reduce the level and then look at what happens to that. But in case of CRISPR-Cas9, it is the genome. You are able to alter the genome. So you can change the bases, you can delete, add, whatever it is. So these are two distinct approaches looking at various aspects. CRISPR-Cas9, you're not silencing just, it is not only for silencing gene, but it's also to introduce some changes. For example, one can do the reverse, like there is a mutation which is causing a disease. This approach can potentially fix the mutation and restore it to the normal form. If that can really be allowed in the human, then you are able to become disease free, technically, right? But in the lab system, that you can use it for any of this. You can bring about changes in the sequence, delete genes, inactivate, things like that. Yeah. Explain details about instruments available for research. I don't know what do you mean by instruments. This is an approach. The rest of the instruments are the routine instrument that you require for any cell biology, molecular biology applications. You need microscope, you need cell culture incubators, you need cell culture facilities, you need PCR machines, and then all these routine, DNA gels, agarose, and all those things which we use for characterizing the DNA and so on. So it's nothing very special about the instrument, but these are more about the approach that you do, right? So I think these are the things. Yeah, with regard to the exams and other things, yeah, you would get to know. I don't conduct the exams, so it will be done by the NPTEL office. Dates, everything will be announced if not already done. But I would recommend you go through your, the NPTEL course portal. You'll get to know. Yeah, right. So that's the the question so far with regard to the thing, and then there are some questions that is coming from the YouTube handle. So let me how to start basic lab for genomics. Wherever it is, where you are going to do clinical research and so on. It all depends on what is that you want to do. Is it something that you want to do diagnosis, or it is for teaching? Is it for research? Then you arrive at what is required. So I wouldn't be able to tell exactly what is that required. So I would recommend that you list down these experiments, what experiments that you wish to do, and accordingly you can come up with what kind of equipments that is required, right? So that's what I would recommend there. Yeah. Where can I do basic workshop for genomics? Well, I mean, of course, these are all questions that we should take offline. So yeah, I mean, maybe last 2.5 years or so, that kind of workshops are not here, but certainly these are workshops are being done in different parts of the country. Yeah, maybe there will be later next year there may be some such program that may be possible from IIT Kanpur. You can look for the websites. Yeah. Lectures will be available even after course tenure. Yes, it is available in the YouTube. All these lectures for all the courses are even available in the YouTube. You can search the YouTube using the term functional genomics and my name. You can find that. I work in the field of pharmacogenomics in my college. That's quite good. So if you're really looking at genetic variations and how the drug respond or how the drug responds in patients, then you can do that kind of study. All you need is um, simple molecular biology equipments like agarose gel, PCR machine, and gel dock and restriction enzyme and so on. You can certainly start looking at it. I mean, that's something that you can really do. Yeah. Okay, so that's about the questions that were there in the YouTube portal and then the Google form. So if there are any questions from the participants who have joined live, maybe you could ask. I would recommend first you introduce yourself, what are you doing, and how do you find the course, and then ask the question. Maybe we can take up a couple of them. You can use your raise hand. Therefore, if there are more than one, we can allow one after the other. Yeah. I'm not sure. Yeah. Anyone, whether you have the access control for asking questions? Maybe Murali, can you please?
Yeah, I think they need to connect it with the audio and whether, okay, so there are a couple of them. So whether they can hear us and if they have good connectivity, they can unmute themselves and introduce and ask.
Yeah, if you're unable to ask, then you can even type it in the chat box like someone did regarding the exam. So
yeah,
even
in
that
fact,
would be.
there is one question from Sarvanan that I would like to name book for references. I'm going to write exam. So more, I mean, sir can add more onto that, but the question paper will be mostly on the topics which are already taught by the sir in the lectures. And for books, there is no specific book because this is something that he has like collected different topics and then it was discussed there. Although we'll be sharing some of the more links which can be helpful for your exam. Yeah, sir can add.
Yeah, I think what Rashmi, what you said is right, and I think for every lecture, in a week after that, we have provided the details, as much detail that is publicly available. We have given you the links and so on. So that's good enough for the exam. But if you are interested in knowing more, obviously you can think of books, whatever that is available. But there's no one book that would cover the topic that we have mentioned here. So, yeah.
Okay, so over YouTube chat box, there is one query from somebody that I'm ISER DSMS student from ISER and whether internship available under you.
Yeah, you can. There is IIT Kanpur offers an internship called Surge. It's a competitive program, and that's the only way you can get into this internship. Those who are interested in should apply. That is for next summer. You may want to see that sometime in January, February. That's when that portal will come up. You apply. It's not just me. There are many more much better faculty in the department. You will get a good internship. Yeah. Yeah.
Sir, good evening, sir. My name is Saravana.
Anyone else? I mean, I don't know whether those who are joined here, whether it's Smriti, Supratik, Shruti, whether you are able to hear us, whether you could ask question, I'm not too sure. Supposedly a live session, yeah. Srivanth.
Yeah. So there is one query in the chat box here in Zoom, that how multiple sequence alignment is used for detecting evolution of a particular organism and provide a better protection against diseases.
What is it? Provide better protection against?
Diseases.
Oh, I see. Okay, there are two different aspects. See, when you look at the genome sequence, when we try to align them and compare, what perhaps you will be able to get is that which species came first and which likely the species came later, more recent, and so on. So you can place them relative to one another in terms of time scale. That's what normally people do, looking at what are the new variants that are present, which is not present in other, and so on. So that's normally done. Okay, that is just to suggest or predict as to what had happened in the past. Okay, that's the prediction. Most likely, the relative positions of the species, perhaps we are able to keep them properly or more correctly, but beyond which it would be difficult to say what had happened. Was there anything missing link that survived but no longer present, and so on? That's difficult to say. But with regard to the second part, whether you'll be able to prevent against any disease, you have to understand that, like every species, whether it is a plant or animal, each one of the species is really trying to survive and leave the next generation. Whether it is a host or is a parasite, both of them are trying to do the same thing. So when you talk about disease, the disease is, if you're talking about infectious disease, and then there are pathogens that infect host and cause disease. And these pathogens are also species. They're also trying hard to survive. So the changes that happen to the genome, that gives a better control for them to survive, isn't it? So, and then therefore, sometimes they win against the host, so the host die if the disease is severe. But the win-win situation always is that the infectious a organism, which could be often microbes, becomes neutral in the sense that they don't really cause death to the host. It doesn't cause really that kind of a symptoms that the death that this host is unable to survive. Because if the host die, even the infectious organism is also at loss, because it can't survive without a host. So what happens is that the changes that happen randomly, some of them lead to a situation where the host is not really affected greatly by the infectious agent, therefore both of them able to co-survive. This is what happens. We have in our body millions of microorganisms that they don't really damage you or do any harm to you, because they have went through this kind of changes. Now they are settled, right? So that's what happens. So it's all something like happens randomly. Some are selected, some are selected against. So looking at the genome and saying that it would we'll be able to predict or prevent the infection is difficult, because we don't know what are the pathogen and what kind of genetic changes they may bring in. So we never know. So only if you study that, we can address this question. Yeah. So any other anyone else? I'm wondering whether all of you who have joined here are able to hear or not. Yeah.
Yeah, so yeah, I see that even many of new ones have joined. We are here. Okay, so Sarvanan has confirmed we are hearing. So it
Okay.
would be nice, Sarvanan, if you can unmute yourself and you can, because this one was one-to-one interaction. So
I don't know whether you have that access control.
yeah, I think they can unmute by themselves. Murli can help.
I don't
Yes, Rashmi, they can unmute themselves and
Right.
ask their question.
Yeah.
Yeah, if there are any questions, maybe some of you can unmute. Yeah.
Sir, good evening, sir. Am I audible?
Yes, you are. Maybe you want to be a little more louder. Yeah.
Okay. Thank you, sir. Yeah, just I'm working in IT industry for 18 years. So basically, I'm doing for medical device product engineering. So and along with past 1 year, I started doing MSc Bioinformatics by Karasmo from Bharatiya University. So I think this course is much useful, and whatever course you are studying in Bharatiya University is like the same subject you are in this course also. So what I want get advice from you is like I want to pursue my PhD in this area as a part time. So what and all I need to focus. So basically, though I am from computer science, like I want to shift towards the biological because bioinformatics, like I found the potential growth and also much useful for human being for upcoming years, especially gene therapy and then finding the cancer mutation and vaccination. So it will be very grateful if we could give some very high available things which I can focus so that it would be useful for my research.
Yeah, sure. I mean,
Any
it is
topics like any basic.
yeah. You know, I mean, this PhD topic is something that is a long drawn affair. It doesn't. One has to really have certain questions, and for you to have a questions, you have to read the literature and you know where are the gaps and whether that gaps can be addressed by any of the means that you think using your skill sets, and you have to find um, a mentor and institute and university to do. So that's the process. So that is something that we cannot really discuss as to which topic that you should look at it. But certainly, I see a lot more possibilities since you being someone from computer science background. I mean, there are wealth of data. I mean, any day, if you go to the website of the National Center for Biotechnology Information, NCBI, just open and then look at what are the database that it has. And every day, every second, people keep adding data to that. These are genome sequence data, there are transcriptome data, there are protein data bank, there are variation, the genome variation data bank. I mean, it is for you to decide which database that you want to analyze and what questions that you're asking. So it's the sky is the limit. I mean, I would certainly recommend that you do look at it and then see what can be done. And you have to have, of course, a mentor who would tell you how to interpret the biological data, and then what kind of tools you can use, and that's a win-win combination. So certainly you should consider that seriously if you are interested in research in this area.
Yeah, thank you very much.
Anyone else?
Yes, Sai Shri or Smriti, Supratik, Shruti, and then Shrivan, R. Thakur. I don't know whether they are able to hear.
Well, if there are no other questions, we may conclude. I don't know. Yes, Saravanan, yeah, you can ask questions. Recent COVID vaccine, yes.
Sir, just my basic question. Like now we have identified Reddit sphere is the protein inhibitor for stopping the virus growth. So like what I want to know is like how we are coming to conclusion like this medicine would be the stop the virus growth. Like if we want to know those kind of things, which and all we need to go through, or what basic things we need to find and read, it will be helpful. So each now we have like H1N1 is spreading. Now they are giving the anti-flu. Like each virus has its own medicine. So how industries like they're building this, which basics principle they're applying. Whether we need to proteomics is useful for us, or genomics will be helpful for us. How they are arriving this medicine is the one which will cure the disease or like spreading the disease in human body. So
Yeah, I mean, just to,
which kind of
Mhm. Yeah, just to give a brief answer for your questions, there are two aspects to your question. One is a treatment, one is understanding the virus. Okay, and the virus variant that we are talking about, whichever name, there may be many new virus variant that may be coming in, and these are viruses that have got mutations in their genome. So that's why they don't behave exactly the same way like the previous variants or other variants that we know, right? And whatever the treatment modalities that people have used, that is dependent on the symptoms, and people have used a variety of combination of drugs to treat the symptoms. Basically, what we do in any of the treatments. So the virus comes, and we know that 14 days, 15 days, if we are able to survive, then the body takes care. Okay, this is basically that's what it is. Sometimes the symptoms are very mild, like you may just have the fever, cough, and so on. So that, if you have something like a paracetamol, probably you're able to bring down the temperature, and cough another thing. If you leave it, survive it with for 7 days, then it disappears, just like any other common cold or flu and so on. Now, for some, it becomes more severe. That is where you require hospitalization. That's where there are treatments that are people are looking at it. So when people treat this kind of patients, they say that this combination of the drug or this kind of therapy works. That's being shared, and that becomes the standard way of treatment of patients. Okay, this is how the clinical treatment is kind of carried out everywhere. Now there are country-specific variations in terms of what they use, because it has its own, each doctor has think about what could be the best options. When a new variant comes, this is something the world, whatever we have tried out, we know that that's good, that's not going to, probably that's not working. If only then people say this is far more virulent, this because it cannot be treated this way and so on. If it also can be treated in the old way, then there's not much of a public panic, right? They don't talk about that in this channel. And then within some time, people, someone identify how to do, and that information shared, we are able to take care. So in this scenario, other than this sequencing and finding out whether the particular variant of the pathogen that has infected is a different variant or old variant, right? That's where the molecular biology, sequencing, everything comes in. Other than that, none of this technique that you have mentioned, proteomics and all, it doesn't really help. Most of this technique that what you're talking about is to understand how the virus infection may change, your physiology. That's where that kind of work, and that's more of a understanding how infections happens and so on. Because that once for example, how does virus enter the cell? What are the receptors onto which it binds? Which is the epitope or which is the receptor? And then you are able to bring in some inhibitors for that. You prevent the entry and so on. So that's the treatment. Okay. Well, vaccines are preventive measures. Basically, you expose the body to a, a variant which is dormant variant, or some, what do you call as a host virus which expresses some of the COVID, for example, SARS virus surface proteins. Therefore, your body thinks that is, then it generates its own antibody and keeps it ready. So when you have an infection, real infection, you are able to respond quickly with antibodies that are already identified that can be produced faster. So I hope I'm able to make it distinction as to which technique can be used for understanding the disease process, which can be used for diagnosis, and which can be used for prevention.
Thank you, sir.
So there's one question. There's can we have a few specific topics of current research on plant functional genomics in India? Well, there are. Obviously, not only in India, that everywhere people are trying to do is that one that you want to increase. Like for example, you're talking about any commercial plant, like for example, was the wheat, rice, or whatever other millets and others. Can you increase their production? Can they be drought resistant? Can they be, for example, um, resistant to any of these infection? For example, the what do you call as Murali, can you mute this Vinita thing? Murali, can you mute this? Yeah. Okay. So one of the common thing which happens in the crops is that nematodes infect the plants, and therefore the plants don't survive, they die, or they become stunted growth, they don't produce as much as it happens in tomato, it happens in any of this plant that you look at it. So because these nematodes live in the soil, and when these are nematodes, I'm sure all of you know, these are small worms that live, and then they can get into the plant and then they can infect them and so on. So any of these, like for example, tomato or anything that you talk about, even legumes and things like that. So what people are trying to do is to, make transgenic plants which would express the siRNA kind of, duplex against the nematode specific genes. So basically you're not making any protein, but you're making certain RNAs. So once the nematode enter the plant, then this RNA would silence the nematode gene, and therefore the nematode cannot really proliferate. It cannot reproduce. So that prevents the infection. So regardless how many nematodes enter, then their infection rate, is reduced. Therefore, the plants can grow now better and give better yield. This is one. And of course, you have come up with many other. This is something which is there in India. There are projects that happen, but also there are people are talking about other varieties also. Even there were projects that talks about you have this essential, non-essential amino acids. So essential amino acids, something that you need to get it from, your food, and then there are, the commonly available food that any low-income group can get, and whether we can enrich these amino acid in this, this is something that people think of it. And of course, you must have discussed or must have heard about the cotton and how some toxins are expressed in cotton plant, therefore that will not be infected by pests and so on. So these are the approaches people take. Basically you want to know, or you want to do something that where you increase the yield or make it disease resistant, or after post harvest, for example, any of these vegetables, if you can keep them fresh for a longer time, because they undergo a process. Basically they, for example, tomato, you keep it longer time, you know what happens. Basically that ripened fruit has to collapse, and then seeds should get exposed, and then they should grow. That's the process. But can you prolong that process? Or even banana, you have seen that. So these are other attempts people try to make. Therefore, there could be benefit to all of us, because if you can make these fruits and vegetables to live longer after harvest, then storage is easier. And if you can store, then the price would go down, obviously, and more people can have it. Otherwise, they are wasted, right? there any query here in this, Rashmi?
Yeah, so now in Zoom chat, there is one query that how pedigree analysis and phylogenetic data be used to address a particular query regarding a specific disease and its treatment. When these analysis becomes essential.
Well, I think these are two different things. When you talk about pedigree analysis, is that you're talking about 2 or 3 generations, 4 generations, which happened within last 60 years or something. You have individuals, for example, your parents, your grandparents, and your next generation. This is a 4th generation. So these people are alive. You can take their DNA and analyze and characterize. That's the pedigree analysis. When you talk about phylogenetic, this we are talking about thousands of years before what could have happened. Like we are talking about how humans evolved and what is the relation between human and chimpanzee, other monkeys, other mammals. We are talking about millions of years, sometimes thousands of years, sometimes. So that's that's the scale that we are talking about. These two cannot be really combined.
Yes, so I think you have taken all the questions already, and we don't see any more question in the neither in the YouTube chat box or not here, and also the ones who are online, they are also not asking. Yeah. So recent question is that could you please explain about the nomenclature of the genes, sir? So I think gene nomenclature, there is a there are proper nomenclature guidelines which one has to follow. So for example, if it has to be written in italicized manner, and for human it has to be in caps. For mouse, first letter is caps and then is small. So there is a complete guideline is there. So you can go and have complete look into that. That would be better. Sir can.
Yeah, that's right. So there are two aspects to nomenclature. One is that how do you give names to genes, right? The other one is how do you represent them. So earlier people used to give genes the names the way they like to, right? For example, I have a pet a dog which is named called Chitty or whatever, and then I found a gene, I can give that name. So that used to be like that earlier. But then what had happened was then there are a lot of confusion. Two different genes can be named with the same name because you are doing independently by different groups. So there was a consortium formed which came up with guidelines as to how the gene should be named for the vertebrates and likewise. So there are detailed descriptions. So if you have identified a gene, if you want to give a name, so you have to write to that consortium that this is what I am proposing based on certain guidelines. So if you have identified a gene for a particular disease, so that gene name would have those elements of what is the disease name, whether it's the first gene, second gene, third gene, and so on. And then they would approve and they'll modify it based on certain guidelines that they are evolving. And then they'll tell you, okay, this is what the gene name is. And that is then it's called as approved gene. Once it is approved, then what Rashmi said is correct. Then for the humans, always you use all letters in capital. If it is mouse, rat, any of the vertebrates, then if the first letter is capital, then rest are all small letters. And then the gene is italicized and the protein can be, the same gene name or but not italics, or protein can have a different name that is also allowed. You can say X gene is coding for Y protein. So that's okay. So that's fine. So for protein, there are no such clear restrictions, but gene names do have certain restrictions with regard to how it can be named here. Fine, I guess we have had fantastic discussions. There are questions that are asked live, and there are questions that were asked in the chat box in the YouTube, and I'm sure there are many who are watching the YouTube as well. And then you have this Google form where there are questions. We have answered most of them, but you may continue to ask questions in the forum. So they are active until a certain time, and I'm sure all of you found this course useful, and you would do well in the exam. And more than the exam and mark you get, it is the understanding, the concepts, what happens in this field. If you can keep up with that by reading popular articles and so on, that is something that we'll be very happy that we are able to introduce you to this topic. So with that, we'll close, and all the best for your exams, those who are taking exams, and yeah, wish you all great in days to come. Yeah.
Thank you.
See you, everyone. Already we can close it now. Yeah.
Yes, sir. Thank you so much, sir, for your valuable time. Thank you, Rashmi, for joining in.
Yeah, thank you.
Participants. We end the session now. Thank you.
Thank you.
Bye, bye, sir.